Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Good afternoon, everyone, and welcome. My name is Kim Ricketts, and I manage, along with Kirsten Wiley, the Microsoft Research Visiting Speaker Series. Thanks for finding our lecture room today, not our usual one. Today we welcome Marco Iacoboni to Microsoft Research to discuss some of the most interesting developments that have happened in neuroscience in a century. Within the past decade, scientists have found the neurons in the brain that provide a plausible explanation for why we can read each other so intimately, and in fact why we are driven to do so. The discovery of these mirror neurons has opened the door to a wide range of experiments and research that suggest our brains are capable of mirroring the deepest aspects of the minds of others at the level of a single cell, in fact, and we do it effortlessly and continuously. This discovery will have groundbreaking repercussions in many fields, from education to marketing to ethics to politics and much more. Dr. Iacoboni has been involved in these experiments since the beginning, barely 20 years ago, and is here to walk us through these experiments and others that have led to the way in studying mirror neurons. He is currently director of the Transcranial Magnetic Simulation Laboratory at the David Geffen School of Medicine in Los Angeles. So please join me in welcoming Dr. Marco Iacoboni to Microsoft Research.
Thank you for your introduction. I must say that you stole the whole lecture. I don't know what to say now. All right, let me give you a little summary or overview of what I want to discuss today. I want to start really with the discovery of mirror neurons. There's plenty of stories on how these cells were discovered. And in fact, even the way the cells were discovered, I think, tells us a lot about how we perceive things, how our assumptions sometimes make us blind to phenomena that are under our nose. There are stories that, in fact, are probably not so true on how these cells were discovered, and I'll tell you how, in fact, it took scientists a lot of time to figure out that they had this phenomenon under their eyes. There are these stories in which people say, well, one day one scientist grasped a peanut and there was a firing of the cell, and there was this immediate realization that there are these cells in the brain of the monkey. Turns out it didn't actually go that way. It's a much longer, more complex process. And then I also want to say in this initial part of the talk what these cells really do, because there is a kind of a simplistic idea of what these cells do. I think that in the media, in the general public, some of the main features have been captured, but these cells are much more flexible and much more interesting than what people think they are. And so that will be the initial part in which I'm going to talk about the monkey findings, the experiments done in the monkey brain. And then I'd like to move on to the work that we have done in the human brain, mostly using brain imaging. And the first thing we thought, okay, well, if these cells really fire when we make an action, when we see other people making actions, of course it seems that they have nice properties for actually being very important for imitation. And it turns out that imitation is really a fundamental way in which people learn and even connect. There is a psychologist from Holland that says that imitation is a social glue. If I start imitating you, suddenly you're going to like me much more than if I don't. There is plenty of connection that just the bodily movements that we do together really get us together at an emotional level. So we started really the work in the human brain looking at imitation paradigms using brain imaging. And I want to tell you a few things about that. But then we also thought, well, if these cells are really fired up when we make actions and when we see other people making action, every time we act, there is an intention behind the action. And so we thought, well, could it be that what these cells really code are not really just the actions of people, but the deeper mental states associated with those actions? And we have done some studies that I'm going to cover briefly, in which we really demonstrate that these cells and this system in the human brain, what it does is to code the intentions of others, which is something that for centuries in philosophy of mind has been considered really a difficult problem to solve. In fact, it's called the problem of other minds. how can we actually access the minds of others if we know only our own mind? And it seems that evolution has devised a very simple mechanism that let us understand in a very simple automatic way the mental states of other people when we see them involved in their everyday actions. But then, if we are so good at getting into the minds of others, can we actually even feel what other people feel? And so one of the claims I'm going to make today is that evolution has again shaped a system in our brain that makes us wired for empathy. And I think this is really a major revision of long-standing beliefs. The classical idea is that biology is all about self-preservation, all about individualism. And we become social animals with our higher order ideas, with our intellect. It seems that we are actually built biologically to empathize with others. And I'm going to show you some empirical data that support this concept. And if we are really good at feeling what other people feel, I think that the distance between self and other, that it's one of the major framework, especially in Western societies, that individuals are all detached from other people. Really, self and other, to me, they get much closer. That's why my slogan is what mirror neurons really demonstrate is the intimacy of self and other. Self and other are much closer than we think they are. And I think that's, again, an important message, especially to send out, because we are so good at dividing ourselves from other people, especially using our beliefs, political beliefs, religious beliefs. But if we really look at deep inside how our brain is organized, it seems that we're really so attuned to each other that if we pay attention to our biology rather than our beliefs, I think our society will be much more empathic. And of course, one thing that we started speculating, and then we have data that demonstrated there is a link between this system and disorders of social behavior, is the idea that there are broken mirrors. Sometimes this system doesn't work at its full potential. And what happens in individuals that have hypoactivity in these brain areas. And so we had this idea that disorders of social behavior may be linked to a hypofunctioning of this system, and we have evidence for that, and I'm going to show you that evidence. The nice thing and the nice implication about this work is that some people are already using our constructs to build interventions on individuals that have problems in social behavior that in fact can help these individuals. And I was talking to Sally Rogers at UC Davis a few months ago, and she's starting this program on using imitation as a form of interventions. And it turns out that she's telling me the results are just amazing. They are preliminary results, but they're really exciting. So that's very exciting because I tend to do a science that is fascinating, but it doesn't have a lot of immediate applications. And in this particular field, it seems that in a few years we went from the discovery in the monkey to a real application in humans. And the final point I want to make, and that's also the way that the book ends, is that if we really revise all our ideas about our biology and our intellect and how we are, and we understand our condition better, I mean, it really seems to me that this tells us that we can use neuroscience and any other kind of scientific discovery to really make a strong change in our society. I think that the message where it's coming out of the lab is that, in fact, again, we are much more empathic than we think we are. And this, I mean, if the moment this message gets outside and we become really aware of this, I think it's going to also change the way we think about a lot of things in our society. So that's the final message of the book, and it's a message, of course, of hope and optimism. All right, I tend to talk to fellow neuroscientists. So I have this tendency to get sometimes, scientists, what they do, the practice of science is really getting into the details of science. I'll try to not to forget that almost that I'm talking about the brain, but I have to tell you something about the brain. Of course, when I try to explain to you how this discovery was made. This is a figure of the primate brain. It's a macaque brain, and the areas that you see in color are areas that have cells that have to do with actions. Every time the monkey makes an action, some of these cells fire up. And by studying these areas, we have a better understanding of how the brain controls movement and our own actions. And the group of Rizzolatti was studying cells in F5, which is a little brain region in the ventral premotor cortex. And in this region, they were studying cells that fire for grasping. Now, grasping is a fundamental behavior in our everyday life. Try to get out of the house without grasping the doorknob. We grasp things continuously. If I want to highlight this region, I use a laser pointer. What do? I grasp it. So grasping is really something that we do continuously. So it's very important to understand how grasping works at the level of the brain. And of course, they weren't expecting at all to find these cells. Again, the story that you often read in newspapers, in the media, is that, well, they were studying these cells, there was an electrode in the monkey brain, and one experimenter grasped something, and the scientist heard the discharge of the cell. And there was this immediate realization that mirror neurons were in the monkey brain. There is another story that is very popular. Actually, I was telling this story in my seminars years ago. And this is a really cute story, and it involves Vittorio Gallese and Italian ice creams that are notoriously very good. So the idea was that during a break from an experiment, he goes out of the lab, and then he buys an ice cream, and he's still working on the ice cream when he gets back into the lab. The monkey's sitting on the chair. There is an electrode implanted in the monkey brain, and every time Gallese licks the ice cream, there is a discharge of the cell. And Gallese immediately realized that these cells fire not only for the action of the monkey, but also for the action of other people. So when I started writing the book, I thought, well, I tell these stories, but why don't I check if these stories are veridical or not? And I called my friends back in Parma, and they told me, you know what, I don't know where these stories start, but these are all urban legends. I mean, we actually, at some point, we had a progressive realization that something like that was going on. But we can't even remember this sudden insight, because even them that were really open-minded neuroscientists, the assumption was that in the areas of the brain that control movement, those neurons don't respond to visual stimuli, and especially don't respond to actions of others. So anytime there was some kind of a discharge in association with the actions of the experimenters, they would just dismiss that, or they would consider this discharge a very complex visual motor response. They weren't really saying, oh yeah, it's coding the action. It took them years to figure out what was going on. And that tells you a lot about the assumptions that we make in science. I mean, you got to make some assumptions, because otherwise the reality that we try to study is too complex. And so we have to have some a priori assumptions, some hypotheses. But sometimes these assumptions really make us blind to phenomena that are just in front of us. And so it really took them a long time to figure out that these cells fire not only when the monkey grasps an object, but also when the monkey observes somebody else grasping an object. At some point, they had the realization, and then they decided to do a series of control experiments to really figure out if that was the case, if there was something funny going on, and if there was some kind of an artifact. Well, 15 years later, or 20 years later, we know it's not an artifact, and we have learned a lot about these cells. And so what I'm going to tell you is some of the properties of these cells. So the basic finding is the following one. These are neurophysiological data. This is called raster. Each one of these lines and each one of these little marks represents an action potential when the cell fires. And at the bottom, we have a histogram that tells us the firing rate change over time. And so these are a bunch of trials. And so every time on the right side, when the monkey grasps, the cell fires very strongly, and you see this robust firing rate change. But also when the monkey observes somebody else grasping, and the trials here are aligned at the moment in which the monkey sees the human grasping the object, you also have a robust firing rate change. So the cells really seems to be coding for both when the monkey does and the monkey simply sees. In fact, one of the early names for these cells was a monkey do, monkey see cell. And so that's, I mean, and you know, the name mirror neuron really captures well this concept, and it really captures the fact that, in fact, we tend to imitate each other and to mirror each other all the time. But on the other hand, it's also a little bit limiting, because these cells are more complex than that. So, of course, they are cells that are specialized for actions. They don't fire, I mean, some people are complaining that we think the mirror neurons do everything. One thing you have to remember is that, for instance, they will not fire if you present just an object in front of the monkey. If the monkey doesn't see an action, there is no firing of those cells. So they're really specialized and seem to be really concerned with actions of the self and of other people. But not only fire for the same action, but often for complementary action. There is 1/3 of cells that are called strictly congruent mirror neurons. They only fire for the same action that they code from a motoric standpoint. But 2/3 of those cells are actually, we call them broadly congruent mirror neurons, because they fire also for actions that are complementary or that achieve the same goal of the action that is coded from a motoric standpoint. And if you think about it, it makes a lot of sense, because human interactions or interactions between individuals of a society can't just be based on mirroring. If a baby cries, you don't want the mother to cry. You want the mother to do something to help the baby. And so the coding of complementary action is really very important in order to facilitate social interactions. They also fire for hidden action. Sometimes you see somebody busy with some activities, but you don't see the whole action. You see only a part of it. And it turns out that there are nicely controlled studies that show that if you see the initial component of the action and you know that, for instance, behind the screen there is something to be grasped, the cell also fires. So it doesn't have to have the complete action in sight in order to code that guy is doing that thing. In fact, the original scientific paper that published these findings, the title was, "I Know What You're Doing." They also fire in complete darkness or without the sight of an action for sounds associated with action. And that's also very important, because often our actions are associated with sounds. We are able to recognize the footsteps of our colleagues when they're walking in the hallway. And it turns out these cells fire, the study weren't done on footsteps in the hallway, but they were done on sounds like, for instance, breaking peanuts or tearing paper and so on. All these sounds drive the response of the cell, even when the monkey is not seeing the action at all. And that's, I think, a remarkable property, because it tells us that these cells really have a fairly abstract coding of the actions of other people. They fire for the action of self, for the sight of an action, for complementary action, and even for action sounds. So they really have a kind of an abstract, the same very cell. So at the level of a single cell, you have a really kind of abstract coding of actions of others and of self. And the most popular ones are cells that fire for grasping and hand actions, and of course they are very important for communication. We use our hands to when we talk all the time. There's plenty of evidence, in fact, that people, even over the phone, when they know that nobody's watching them, they still use gestures when they talk. And there are linguists that say that gestures and words are part of one system, which is language. Of course, Italians use it much more. But there are also cells that code for mouth actions and facial movements. And I think that's very important, because when we get to the aspect of empathy, we want to remember that, in fact, we transmit our emotional expression very often with our face. So this is a study done by Francesco Ferrari. He's an interesting guy, because he got his PhD working with a scientist that studies monkeys' behavior. And after his PhD, he did a postdoc in Rizzolati's lab doing single unit recordings in the monkeys. And you can tell that he studied behaviors of monkeys very well, because he's really good at doing these kind of gestures. And he studied ingestive gestures like biting a banana or drinking juice from a syringe, or that's the way juice is delivered in monkeys that live in the lab. They get juice through the syringe, and they love juice. So for them, it's an important tool. And also communicative gestures. This is called lip smacking, and it's a facial gesture of positive valence in monkeys' social communication. So there are mirror neurons also for these facial gestures. And I think that those are important when it gets really to the point of trying to figure out how these cells implement empathy, understanding the feelings of others, because we use our face a lot in order to convey what we feel. Well, at some point we thought, well, it was really early on. They had just published the full-blown paper in '96 on mirror neurons, and Rizzolati asked me to be part of an international. I was already back here in LA, down there. A lot of people think I'm from Parma because I'm Italian, and because I've done early work on mirror neurons, I've published papers with Rizzolatti and collaborators. And so, of course, it's a natural assumption. Probably Jacoboni was in that lab, and then he moved to the US. Turns out I'm from the Eternal City, from Rome. And I actually met Rizzolatti when I was already in LA. And I met him in Prague. So he asked me to be part of this international group that wanted to study mirror neurons in the monkey brain, but also in the human brain. I'm a brain imager. I do brain mapping, brain imaging. I use mostly fMRI. In those days, we were using also a little bit of PET. And now we use a lot of transcranial magnetic stimulation, which is a nice technique because it gives you complementary information compared to fMRI. And I'll briefly tell you a couple of studies on that. We're using TMS. But so the idea was that, well, how do we translate? And this is, the part in which I'm gonna try to explain how we kind of think our experiments, and we move from the monkey brain to the human brain. So we thought, well, let's try to translate the findings in the monkey brain with the findings that we can possibly have in the human brain. And of course, we were inspired by the work of somebody who actually lives here, Andy Meltzoff. He has done amazing work on neonatal imitation, and we thought, of course, the properties of these cells seem to be very important for imitation. And Andy Meltzoff has really done an amazing work in demonstrating that we really learn by imitation. When he did this work, the classical, the dominant view in developmental psychology was that babies do not imitate right away, and actually toddlers learn to imitate toward the second year of life. And Andy, what he demonstrated with this work is that imitation really, from the get-go, it's a nice way of bypassing trial and error learning. He also achieved something else. I always tell him, Andy, you're the only guy I know of that managed to publish a paper in one of the most prestigious journals in science with a picture of yourself sticking your tongue out. And so the idea was, let's use imitation paradigms in order to test mirroring properties in the human brain. And let's use the monkey data to try to translate these findings in the monkey brain from the single cell to activity in the human brain. And so one thing we noticed was very early on was that there was a much higher firing when the monkey makes the action compared to just observing it, which kind of makes sense, because if there was a completely identical fire rate change, you would be doing the same thing all the time. You want to kind of suppress the tendency to imitate others. And so we thought, well, let's use this kind of firing pattern and let's predict changes in activity with fMRI. And so we predicted that there should be some activity when subjects make a motor task, a little bit reduced activity when subjects are just observing the action. But when it comes to imitation, when you imitate, you actually do it and see it at the same time. And so we thought maybe there is an additivity of these two signals, such that during imitation there is a stronger activity. And so this was our predicted pattern of activity for areas with mirror neurons in a variety of different tasks, in which sometimes you imitate, sometimes you do it, and sometimes you see it. We did a very simple study. This was the very first study using very simple finger movements. And there were a bunch of control conditions, of course. And we found two regions in the human brain with exactly the pattern we predicted. And these two regions were located in areas of the human brain that seemed to be anatomically very similar to the areas of the monkey brain that contain mirror neurons. One of these areas, it's in the frontal lobe, and it's called, in the left hemisphere, it's called Broca's area. Why? Because Paul Broca, which is a French neurologist, discovered that this neural center, it's very important for language. So there was already a kind of a hypothesis that these cells may be precursors of systems in the human brain that are important for language. And our finding really kind of supported this hypothesis. On the other hand, when you do see activation in a language area, even in a non-language task, you always wonder, well, could it be that this activation is actually due to some kind of verbalization, internal verbalization of the subjects? And so we thought, well, let's use TMS to test the hypothesis that this region is essential for imitation. So again, when we do TMS experiments, we place a coil on the head of the subject, we send an electric pulse, this pulse creates a magnetic field, and the magnetic field it's the surface of the brain which in turn generates an electric flow in the brain, and it kind of injects noise in the brain tissue underneath the coil. And so you can interfere with the activity of those brain cells. We call this going from brain mapping to brain zapping. So we did an experiment in which we actually stimulated Broca's area, and there were some imitation conditions and some control conditions, and we also stimulated some control sites. And what we demonstrated was, in fact, that Broca's area is essential to imitation. When we stimulated Broca's area, these subjects had reduced performance in imitation. And when they were doing a control task, that didn't happen. So the data really demonstrated this region is an essential area for imitation. So right now we think that there are two major centers in the human brain that contain mirror neurons. One is located in this part of the frontal lobe that contains also Broca's area. And another one is in the parietal lobe, which is a part of the brain that seems to be very important for putting together perception and action. And we also know that there is, in the temporal lobe, which is an area of the brain that is really important for visual processing, there are cells that seem to be specialized for coding the actions of others from the visual standpoint. So the idea is that these cells really are the ones that send the visual input to the mirror areas in the human brain. And now I want to talk about the possible role of these cells in understanding the intentions of other people. Sometimes this thing is called mind reading. There is a whole camp. There are actually two major camps in the literature when it comes to understanding the mental states of others. One that says, well, we actually use the similar approach that scientists use when they actually study natural phenomena. We see other people, they make certain, they have certain behaviors, and then we kind of make an inference from their behavior with regard to their mental state, because we can map those behaviors with our own behavior, and we know what are our mental states. So a very complex inferential process. And actually I thought, why on earth evolution might have devised something so complex? After all, we read the minds of others all the time. I'm reading your minds now, and you're reading mine. And we do this effortlessly, continuously. So, we would be totally exhausted after 10 minutes of interacting with other people if we were using this very complex inferential mechanism. Can't we use a much simpler simulation approach, such that we activate the same areas that are activated when we make those actions, when we see other people making those actions, and by doing that, we trigger a cascade of signals in the brain that let us really get to the intentions of others. The way I like to introduce this issue, the inferential versus the simulative kind of way of understanding the minds of others, is by introducing these two guys, Professor Snape and Harry Potter. I was reading the 5th book of the Harry Potter saga a long time ago. And I don't know how many of you have read these books, but in this particular book, Voldemort, which is a mean wizard indeed, wants to get into Harry Potter's mind to figure out some secrets of the Ministry of Magic. And Professor Snape is supposed to teach Harry Potter to protect his mind from Voldemort to get inside the mind of Harry. And so he's explaining this to Harry, saying the Dark Lord, which is Voldemort, is highly skilled at extracting feelings and memories from another person's mind. And Harry says, he can read minds. And at that point, the answer of Snape, after reading all the 7 books, I like Snape much more, especially after the last one. But at this point, while I was reading the 5th book, I really couldn't stand this guy. But after reading what the answers to Harry, I thought, well, after all, Professor Snape is not that bad. Because he says, you have no subtlety, Potter. Only muggles talk of mind reading. The mind is not a book. And that's exactly the point that I'm trying to make with this study. The idea is that especially for everyday simple actions, you don't really need to get into the symbolic level. You can use this very simple simulation and mirroring that you have in your brain to understand what other people are thinking. And so we did an experiment that it's been often cited in the media, and it's been dubbed the Tea Party experiment. And the main idea is that you often I mean, there is not a one-to-one mapping between actions and intention. I can grasp this thing because I want to drink, or later on because I want to clean up the speaker's desk. So the same grasping action is associated with different intentions. And we cannot devise an experiment because, you know, what we do are experiments. So we really have to try to reduce a complex reality in something that is manageable and can make predictions. And so the idea was to embed a grasping action with different kinds of contexts. In this particular situation, the context suggests somebody is going to drink tea, having some kind of a tea party. Everything is neatly organized. There are cookies, there is a teapot, and so on. We made little video clips that we presented to subjects. There was also a little Italian signature in this because there was a little Nutella right there. And then we used the same set of stimuli, but now the context really suggests cleaning up. There are cookie crumbs, dirty napkin, this is empty, even the cup is empty. So the grasping action is the same, but the context behind it is different. I'm showing you two different grasping actions because what we tend to do when we grasp is to grasp either by the body of the cup, that's called whole hand prehension, or grasping from the handle of the cup. This is called precision grip. Turns out that this is the way we tend to grasp in order to drink, so we didn't want to deal with this problem, so we counterbalance the number of these kind of grasps and the number of this kind of grasps for both contexts. So this experimental design makes a very simple prediction. If the areas that contain mirror neurons are only interested in the grasping action, they're going to fire equally, or they're going to be activated equally for both conditions. But if they're actually coding the intention associated with the action, they should probably also be activated differently. And ideally, you want to see higher activity for drinking, which is a much higher, much more primary intention than cleaning up, especially if your subject are college students. And in fact, that's what we found. The classical mirror neuron area, where we think mirror neurons are contained in the frontal lobe, there was a differential activity for watching grasping actions embedded in this context that suggests drinking compared to this one. And this really tells us that, in fact, what these cells do is coding the intentions of other people rather than the actions of other people. After we did this brain imaging experiments, the monkey people back in Parma did a similar experiment in the monkey, and they even demonstrated that at a single cell level, you have coding for the intention rather than for the action. This is one of the few cases in which brain imaging actually inspires the monkey people to do a study. It's generally the opposite. And one of my good friends, Vittorio Garlese, he knows a lot of French phenomenology. He told me, well, you know, you should really read Maurice Merleau-Ponty because he has quotes that are really ideal for these kind of studies. And in fact, I mean, I found one in one of the major books of Merleau-Ponty, Phenomenology of Perceptions, that really seems to be written for this experiment. It is as if the other person's intentions inhabited my body and mine his. It really seems that with these cells in the brain, it's very simple for us to get into the deeper mental states of others, what they want to achieve with their actions, the goals that they want to achieve, the intentions associated with those actions. And then we thought, well, okay, let's get then into the domain of emotion. If it is possible to understand the intentions of other people in very simple actions, everyday actions, can we feel what other people feel when they express their emotions? There's plenty of authors in the literature that have actually already described this in very powerful ways. I think that the oldest one I found was Montaigne in the 16th century. But I want to actually quote two people. Wittgenstein, in one of his later works, he says, "We see emotions. We do not see facial contortions and make the inference that is feeling joy, grief, boredom. We describe a face immediately as sad, radiant, bored, even when we are unable to give any other description of the feature. We have this automatic way of really understanding, of capturing that face expresses happiness or sadness and so on." And Adam Smith, a long time ago, when we see a stroke aimed and just ready to fall upon the leg or arm of another person, we naturally shrink and draw back our leg or our own arm. And when it does fall, we feel it in some measure and are hurt by it as well as the sufferer. This is a really nice description of what empathy is about. And in fact, the idea here is that if you really have a good introspection, you really see in yourself already that you have this tendency to do these things. So the idea was, how do we actually get mirror neurons involved in all this business of emotion? Because after all, these cells are not really located in areas of the human brain that are known to be concerned with emotions. And there was also another link, a link between empathy and imitation. There is a phenomenon called the chameleon effect. This is a widespread phenomenon. It turns out that pretty much everybody, when they interact with other people socially, they tend to imitate each other. This phenomenon has been studied by social psychologists, and you can actually quantify the amount of imitation that people produce. And some people are more chameleons than others. And it turns out that the more you tend to be a chameleon, the more you tend to be empathic. There are a nice correlation between the tendency to empathize with people and the tendency to imitate others automatically when they interact socially. I wanted to capture this visually because, pictures are always much more powerful than words. And we found these two pictures involving former President Carter. He's given a speech, and this was the chief of his staff at the time he was president. Later on during the speech. So this tendency to imitate others correlates nicely with the tendency to empathize. And so it really gives us, from a behavioral perspective, the idea that in fact these mirroring cells may actually interact with emotional brain centers in order to provide empathy. But as a neuroscientist, I always have to deal with the anatomy of the brain, because signals don't get transmitted just by magic in the brain. And it turns out that I was looking into the anatomy of the primate brain, and of course the emotional brain centers are very different from the mirror neuron areas. But I found that, at least from the anatomical connection that were described in the literature, there was a region of the primate brain, the insula, which is a tiny interesting region that connects mirror neuron areas with the emotional brain centers. And so I thought, well, maybe what happens when we see a happy face and we immediately kind of feel happiness and understand what this person is feeling is by using this relatively simple mechanism in our brain. First, we simulate the facial expression with our mirror neurons. And by doing that, we then send signals to the insula, and through the insula all the way to the emotional brain centers. And here we feel the emotion that other people are feeling. So we did a series of studies to demonstrate that with brain imaging. And we in fact demonstrated first that there was a system in the brain, in the human brain, in which these three major centers go together when you observe and when you imitate facial expression. The activity in these areas are all kind of synchronized. And again, this kind of reminded me of another quote from Merleau-Ponty: I live in the facial expression of the other as I feel him living in mine. This was kind of a proof of concept. These three regions go together and they are activated with similar degrees of activation in a variety of situations in which you observe or imitate facial expression. But it really didn't demonstrate that these systems really maps onto the ability to empathize with other people. So we did a series of follow-up studies. And one thing we're doing now, it's a study on pre-adolescents, and we follow them up through adolescence, a very turbulent period. This is my daughter that posed for me when she was 9. She's now 12, and I bet if I ask her to pose for me today, she would say, why would I do that? She's already a full-blown adolescent. They're imitating and observing facial expressions, and we use different kind of faces so that we can also study the effect of gender, of race, and so on. But we have just preliminary data. We have just looked at the overall effects. And what we found, in fact, the whole circuitry, these mirror neuron areas, these insula, and the emotional brain centers in the amygdala, what they do is not only activate as in the adult experiment we did some years ago, but they also correlate with the ability of these kids to empathize and to be socially competent. We have two measures of social competence. One is called the interpersonal competence. It's a questionnaire that the parents filled up, and they tell us how many friends the kid has, how many play dates he gets every week, how popular is this kid. And there is a correlation between the popularity of the kid and the activity in these areas when these kids do either observe or imitate facial expressions of other people. And we also have a correlation between the empathy scores of these kids, how much these kids are able to empathize with others, and the activity in these regions. So we really call this system a biomarker of society because it really seems to correlate with social behavior of people. And that's why I also say, that these neurons really tell us something about the intimacy of self and other. I think there are these ideas that the self and the other are so detached, so separated. And especially in the Western culture. There are some cultures, especially the Eastern ones, that actually, if you read books from those cultures, you realize that there is much more sense of communion and intimacy between self and other. But then I thought, well, if there is really an intimacy between self and other, and if these cells really let us read the minds of others, can they also be important for building a sense of self? And again, most of my work in brain imaging was inspired by really neat work in developmental psychology. There is a study done in children that looked at imitation and self-recognition. The way they do the study is the following. First of all, how do you get the awareness that the face that you see in the mirror is your own face? This is a nice, actually, question that you can even ask experimentally in other species, not just in humans. There is a simple test to actually test this awareness of self-recognition. It's called the mirror test, another mirror in this study. So the way you do it is with kids, when the kid is sleeping or when he's not paying attention, you put a little mark on the forehead of the kid. And sooner or later, the kid is going to be in front of a mirror. And what you're going to see is if the kid is aware that that face is his own face or her own face and sees the mark for the first time, he directs his attention and his fingers to the mark. This experiment was done initially in chimpanzees. And in chimpanzees, it's been demonstrated they're actually able to recognize their own face through the mirror. And kids are able to do that relatively late in development, toward the end of the second year of life. You probably have seen babies and infants looking at themselves in the mirror when they're much younger, even before the first year of life. But if you do the mirror test in those kids, they're not going to show any mark-directed behavior. That means that probably what they see, it's something very entertaining for them, but they don't have the concept that that face is their own face. But toward the second year of life, they actually achieve this feat, which is an important one, to create a sense of self. And there is a study that shows that if you pair kids, if you put them, there are dyads of kids, 2 kids in one room and 2 other kids in another room, and you put together the kids that are able to pass this test, they already have a good sense of self, they also imitate each other much more. If you put in the same room 2 kids that do not have this sense of self, they imitate each other much less. So this gave me kind of the idea, well, maybe what these cells do is not just getting into the minds of others, but through these reciprocal relations between self and other, they're also helping us building a sense of self. So we did an experiment in which we were morphing the face of our subjects with the other faces, and we looked at the ability of these subjects to recognize their own face or the face of other people. So these subjects were looking at pictures that were kind of a blend of their own face and the face of others. And there were different percentages of self and percentages of others. And what we were able to track down was that, in fact, the areas, the same areas that we think contain mirror neurons, were much more active when subjects were able to recognize themselves. We also did a TMS study in which what we did was stimulate these brain regions, and the prediction was if these areas are really relevant to this behavior, then there should be a decreased performance in the ability to recognize your own face in a morphed face. And we demonstrated that too. So it really seems that these neurons are really not just for others, but also for building a sense of self. And I really think that this tells us that self and other are just 2 sides of the same coin, and that self is really a social construct. It's not something that is trapped in ourself and it's unable to reach toward others. So really, this system tells us that we have this ability to mirror other people automatically. We can easily understand the action that they're making, the intentions associated with those actions, the emotions that are associated with the body expressions that we make. So it really makes us wired for empathy, but it also helps us creating a sense of self. But then what happens when you have a dysfunction of this system? One of the hypotheses we had was that there may be broken mirrors in autism, and there may be a hyperfunctioning of this system. Some groups had made some hypotheses early on, and we actually did experiments that used the same kind of paradigms, imitating and observing facial expressions. Our kids were in the scanner, and we had a group of typically developing kids and a group of kids that belong to the autism spectrum disorder. And one thing that we noticed right away is that the activations in the two groups differ dramatically in these inferior frontal gyrus areas, which are the mirror neuron areas in the frontal lobe. So the typically developing kids activated these areas, and the kids with autism did not. And when we make a direct comparison between the two groups, the reliable difference in brain activity is exactly in these brain regions, so in the mirror neuron areas in the frontal lobe. Most importantly, what we also found was that the activity in these areas in the group of patients with autism was correlated with the severity of the disease. We had some measures of severity of disease, some widely used scales, the ADI and the ADOS. And there was a correlation such that the more reduced is the activity in mirror neuron areas, the more severe is the disorder. And people are really picking up this concept and really making interventions that are based on imitation. And Sally Rogers is really the one at UC Davis at the Mind Institute that is really leading this field. And they're doing these imitation interventions that are relatively simple. You can actually teach the parents to do those interventions. And they're really not based on the fact that you tell the children, well, look at me and do what I do. It's much more a kind of a very warm social interaction. Often what the therapist does is to first imitate what the children are doing. And then in this way the child really gets engaged, and they start leading and the child, and they start doing intervention in these children. I gave a lecture some years ago in the Bay Area for one of these autism foundation, and it was really based only on neuroscience, but in the audience there were plenty of therapists. And at some point, one guy, I already left the stage, came to me and told me, you know what you say makes a lot of sense to me. I work with really impaired patients, and sometimes, some days with these patients, it's impossible to do anything. They seem to be, they don't even see me. In my last resort, when I really have no other way of trying to work with them, I start imitating the motor stereotype movements they make. When I start doing that, they suddenly see me, and they immediately interact with me. And there was a video that a French scholar sent me a long time ago, in which you can really see this thing dramatically. There is a patient, a child with autism in a room with motor stereotype, is flapping the hands, and another kid comes in. It's not even a therapist, it's another kid, and she's playful. And Jacqueline Nadel, which is this French scientist that studies imitation in autism, she has all these rooms set up in a way there are always two copies of the same toy or object. And so the kids that come in start playing with the child with autism, and he starts interacting with him, and she offers him a cowboy hat, and she puts sunglasses on, and then offers the sunglasses to the other kid. And the other kids start imitating what she does, and you can see that the motor stereotype movements of this kid immediately disappear. It's almost magic. Then the other kid leaves the room, and the child with autism stays in the room, and within 30 seconds, the motor stereotype movements come back again in the child with autism. So there is really something powerful about imitation and the ability to really create a social glue. And again, Sally Rogers is really kind of exploiting this concept in order to use interventions that are based on imitation. And what she told me was that they do not only get better in the social domain, but they seem to get better even in the language domain. And so there seems to be an improvement across the board. The data are very preliminary, but she was very excited about that. So let me actually try to wrap it up, and I really hopefully you're going to ask me questions about this research. I think that there's plenty of exciting things about this research, and I think that the message that we are getting out of our lab experiments is that evolution selected these cells really to facilitate social interactions. And these cells are really creating a relatively simple way of interacting with other people by really achieving the depth of the minds of others. These cells really solve what is called the problem of other minds in philosophy of mind. Again, the idea was that how can I actually have access to the minds of others if I can only access my own mind? There are plenty of books written on this, and mostly in philosophy of mind, the solution to the problem is a very inferential and complex one, and these cells seem to provide a simulation approach to understanding the minds of others. And again, I think that one of the most important messages is that this really calls for a major revision of long-standing beliefs. We always thought of our biology as something that is just about self-preservation, just about the self, and that we become social animals with our higher order ideas. This research really tells us that we're really wired for empathy, that we really automatically are attuned to other people. And this seems to me it's a major message. In fact, the more I think about it, the more I think it's the other way around. What divides us are our beliefs, both religious and political. I think we should really try to think of the way evolution shaped us to really try to be more empathic with other people. And yeah, I think that what we are really being wired for involvement and care. And so in the final chapter of the book, I call this neuroscience existential neuroscience, because although one aspect of existentialism is associated with dread and despair, there is another one that I think is much more important, which is associated with involvement and care, commitment to other people. And I think that's what our brains are built for, really to be committed to others, to be involved and to care. And the final point I want to make is that, all this stuff, it's always been implicit, okay? Societies are built on explicit discourses, not on this implicit automatic mirroring. But I think the fact that we are now aware that we have this very powerful mechanism in our own brain, and we can actually talk about it, we can reach the explicit level on which societies are built. I think that we can actually use this message to really have a strong impact on our society and make a better one. All right, thank you.
Correlation between neurons and learning, which comes first?
That's a great question. Which comes first is something that we don't know. We have some hypotheses. One thing that we think it happens is that I always give this example. When it comes, for instance, to early development, often people think that the baby is always imitating the caregiver, the adult, the mother, the father, but it's often the opposite. What happens is that adults imitate babies. And so in a scenario I often give is that baby smiles, mother smiles back. In the baby's brain, it's very simple to associate the motor plan for smiling with the sight of somebody else smiling. So you create mirror neurons. You can create mirror neurons with this reciprocal imitation. And when it comes to learning, though, the story becomes more complex, because at some point we have to conceive that in a way there may be mirror neurons even before you are able to perform certain actions. Now, we have some data in the monkey brain that monkeys do not use tools. And so the early studies on mirror neurons have shown that, in fact, the monkey mirror neurons do not fire when the monkey sees a human grasping something with a tool. But sadly, some years ago, about 10 years into mirror neuron research, some scientists found some mirror cells that fire for tool use actions. So this is a cell that fires when the monkey grasps something, but also when the monkey sees a human using a tool to grasp that thing. So it seems that it is possible, although it's hard to prove definitely, that these cells actually acquire the visual property of firing to the side of somebody else using a tool. And so in principle, it is possible that the formation of these cells precede the ability to actually use those tools, and may actually really be important for learning.
So this may be kind of related, but I wonder, is there any research on cross-cultural studies? So some societies that are more individualistic and some of the impact on.
It's a great question. We did one study that we published last year, had a lot of media coverage. It was a study in which there was a community of Nicaraguans in LA that is very insulated. And so we actually wanted to do a full-blown crossover study, which we had Americans and Nicaraguans watching American gestures and Nicaraguan gestures made by American actors and Nicaraguan actors, all counterbalanced and crossed. We couldn't do the full-blown study because we actually couldn't find any Nicaraguan that wasn't already exposed to American gestures. And the kind of gestures we were using are called emblems, like the OK sign or hang loose. These are stereotype gestures with a very fixed meaning, but some of these gestures have a strong cultural significance. And of course, for instance, this one wasn't in the Nicaraguan culture, and I couldn't even do the Nicaraguan gestures because it's really stuff that I've never seen before, and I've seen it when we were doing the experiment. Now I've completely forgotten about it. So we were studying the responses of this system in the human brain when American subjects only were watching American gestures and Nicaraguan gestures made by American actors and Nicaraguan actors. And we found two things. Of course, there was a stronger response in mirror neurons when the subjects were looking at American gestures. That makes sense. It's more familiar. It's part of their culture. But we also had a kind of a very enticing finding. When the subjects were watching the Nicaraguan guy, and they didn't know it was Nicaraguan. They were seeing just two guys with white shirt and jeans, but except that one was blonde and the other one was dark skin, very dark hair. And the dark skin, dark hair guy is doing both the American gesture and these other things that for American subjects are completely meaningless. But guess what? Their mirror neurons were firing more when the Nicaraguan guy was doing the Nicaraguan gesture than the American ones. They were almost kind of getting that there was a better fluency in this guy, or a better kind of resonance between the identity of the actor and the kind of emblem they were looking at.
Do you think that because you are familiar with that gesture, you don't need to fire as many mirror neurons, whereas if there's a gesture you don't understand, you're trying harder to understand it?
They would explain the findings in the Nicaraguan actor only, but across the board, I mean, when we were looking at the American actor, there was a much higher activity for the American gestures. And overall, there was an overall activity, higher activity for the American gestures. So that finding can't really be explained with that.
And have you looked, or has anyone looked into, is there a way to train yourself or chemical to cause you to have higher levels of, artificially higher levels of neurons firing?
Great question too. I think because, one of the questions that we have to ask ourselves, what do we do with this? Of course it tells us a lot about us and how we interact with people, but can we actually change it? Can we make it even better? It turns out that training, even short training, changes these experiments are done with very simple actions, but it really changes the way these neurons or these neural systems respond to different kinds of actions. We have evidence that just being exposed visually to certain actions activates these cells more in the human brain. And if you actually, for instance, a classical finding is that, of course, you fire more when you make an action, say, opening your hand, and you see somebody else opening the hand. But what if I train myself to open my hand every time I see you closing your hand? 20 minutes of this training really changes entirely the response of this system. So you can actually train the system.
Is it possible to reduce complexity of the study by studying simpler brains than like monkey brains? Can you find the mirror neurons in simpler brains?
Well, the original finding was done, in fact, in the monkey brain. Of course, my hypothesis is that in the human brain there are many more of these cells and probably have much more complex properties. For instance, we have a finding that we haven't published yet, the paper is under review, in which we are using a really rare clinical opportunity. There are patients with epilepsy that they need to be operated. And in order to know exactly where is the focus of epilepsy, the surgeon needs to do two things. First of all, to implant electrodes in the depth of the brain of these patients. And second of all, to remove the drugs that control the epilepsy. Because by doing that, at some point, the patient is going to seize. And when the electrodes are implanted, at the beginning of the seizure, the surgeon knows exactly where is the starting point of the disease. And he can remove only that piece of brain. Doesn't have to make a big resection. Well, during these days in which the patients are implanted with electrodes, we can do experiments on them if they allow us to do it. And they invariably do. And what we have found was, in fact, that not only in mirror neurons, in fact, when I showed you that picture, I said the idea is that we have these areas with mirror cells, but it may be that we have to revise it, because that's, the nature of science. Data can make you change your mind. And in fact, we have evidence that there are many more areas with mirror neurons in the human brain. We also have evidence that some of these cells fire in opposite ways. So for instance, they increase the firing rate when the patient makes the action, but they decrease the firing rate. They shut themselves completely when the patient observes the action. This is something that's never been seen in the monkey. And I think that these cells can do two things. First of all, they control your tendency to imitate other people, because otherwise you'll be imitating all the time. But the other one, with just the firing of one cell, they can easily make a differentiation between self and other. So I think that in the human brain, we're going to find many more types of mirroring cells and much more complex properties. But to go back to your question, well, there is evidence that, for instance, in birds that learn how to sing a song, in their circuitry for songbird learning, there are cells that fire when the bird is singing and when the bird is listening to the song. And these are cells that are located in motor structure, because if you make a lesion there, the bird doesn't sing anymore. That seems to be mirror neurons for auditory stimuli and for learning how to sing a song. So if my hunch is correct, that the way you create these cells is by just a simple association between what you do and what other people do or other individuals, association learning is one of the most common mechanisms in the brains of all sorts of species. So in principle, you can conceive that many more species have the capability to have mirror cells.
Do you see any possible connections between this and the work of Paul Ekman, who studies facial expressions?
Oh yeah, I mean, he has shown that we are really good at getting the emotions of others, even quick changes in the emotion of others, very quickly. How do we do that? I mean, if you have to go into this very complex and laborious inferential process, there's no way that in 150 milliseconds you can actually detect changes in the facial configuration of other people. But if you have right in your motor cortex, the same cell that fires when you make the action, that also is activated by the side of somebody else making the action, you shortcut a bunch of intermediate steps. And so it explains the rapidity and the automaticity in which we are able to recognize and detect facial changes in other people.
I was struck by your comments about sounds and the absence of any other stimuli, and seems to have a parallel with also language learning and acquisition. I was wondering, as an adult, to sort of empathise and mirror the descriptions of actions that happen when someone describes them, so you can listen to the radio, someone talking about it. Have there been any experiments done with children learning language? It sort of feels like they learn as much by the tone of the voice of the person speaking as the actual words, and so they may know more about how they learn through that emotional side rather than the script meaning of the words. Have there been experiments which look at that by using, say, an emotionless rendition of the same language?
Well, I don't think that's been done any imaging study, but as far as I know, there are studies that show that, in fact, you want to have a much more prosodic. I mean, the way we talk to babies is to using Pat Cool, which is a famous researcher in brain development here at University of Seattle. She calls it motherese, the fact that we actually tend to imitate the way babies talk to emphasize the prosody, the pitch of our language, because it's not just the meaning, the cold meaning, but all these other information is very important for communication.
I know that you've studied people watching movies and what happens. So have you found a difference between people, being in the same room together, face-to-face, mirror neurons, and people, on screens?
Well, what we have found, I mean, brain imaging is not the friendliest techniques to do very ecological things, because you have a subject in the scanner with a high magnetic field. But we have done studies with very simple stimuli and with much more realistic stimuli. And one thing that you notice is that the more realistic it becomes, the stronger is the mirroring response. In the monkey brain, when we started this international collaboration, there was also a group of engineers that were part of the consortium, and they were supposed to build virtual reality environments so that the monkey people could really study single cell activity when they could really manipulate all the various parameters of grasping something and so on. Well, that was a complete failure. The monkey needs to see the person in order to have the cell to fire. A monitor doesn't activate these cells at all. Even a virtual reality environment doesn't activate the cells in the monkey. I mean, humans can activate this system even just by looking at the screen, but the more realistic it is, the better it is. And I think that face-to-face interaction, being in the same room, is much more powerful than being detached or looking at people on a screen.
Are there any further questions? You're going to be here a little bit longer before we have to sweep you back across the bridge.
Sure.
Home.
20 minutes or
He's
so.
happy to answer further questions or sign books to take home. Thank you for coming.
Thank you.